Um, so the first thing I want to say is that in the case of Xylella, disease and pathogen are not the same thing. I'm sure that Senator Purcell and others have already discussed this special aspect of Xylella, but I just want to remind everybody that infection with a pathogen does not lead necessarily to disease. Okay. Oops. The second one, I think everybody understands also that it's a complex system, not necessarily a plant disease or a vector entomology problem. It's a really complex system, and everybody should keep in mind that understanding of disease and epidemics as a whole really requires integrating all the different components of the system. So I have a couple more introductory slides, and the first one is what do we really think about when we talk about bacterial species. So here, can you see my arrow? Yes, okay, great. So here you have two examples from E. coli, and one here, what you have over here is the number of, let's call them genes in E. coli, that have been found in E. coli genomes. Here you have 90,000 genes total. And here you have the number of E. coli genomes that have been sequenced. And the core genome, the core genes which are shared by all E. coli are in red. So essentially what you can see is that there are a very small fraction of all the genes you find in E. coli, or all these different bacteria that have been sequenced, 2,000 of them, that we call E. coli. The vast majority of genes here are not core. They're not shared by all E. coli. It's about 5% or less of the genes are shared. Um, on this side here, on the right side of this slide, you have about the same kind of graphic with a smaller data set. I think this was about 20 different genomes. And here is the same thing. Here you have the number of genes, and here you have the number of genes that are unique to only one isolate. And here you have 8,000 genes that are unique. And in this data set, you go here to the other end, and you have about 2,000 genes that are shared by all of these different E. coli isolates. Point being here that we call E. coli a lot of different bacteria that are incredibly diverse, ecologically, biologically, and in terms of their genetics and the genome. Yet we call all of these things here the same, regardless of the fact that most of the genome is not shared among all these different organisms. And the last introductory is to tell you that bacterial genomes, since we're focusing on Xylella, is that genomes tend to be very dynamic, and Xylella is no exception. There are different processes that lead to genome reduction or increases in genome size, but for Xylella, one of the most important ones are over here. Xylella has, of course, lots of phage and prophage regions in the genome, which provide a lot of interesting characteristics that affect biology. Xylella has also been shown to have sex, essentially conjugation, exchange plasmids. That has been demonstrated. And Xylella also picks up DNA from the environment. And the fact that Xylella is naturally competent is actually really important for Xylella diversity, because when you have multiple genotypes of Xylella in the environment, they can exchange DNA. And essentially what they do is they replace one allele of a gene with another allele from another genotype, and you end up with a new combination of Xylella, essentially. And that can have, there's a lot of indirect evidence suggesting that that's very important for the emergence of Xylella diseases. All right. So essentially what I'm telling you in these few introductory slides is that Xylella is absolutely no different from any other bacteria out there. There's plenty of genetic and phenotypic diversity that we already know, And we should expect there is nothing new there, except that we tend to think that Xylella is for some reason different, while that's not really the truth. So what I'm going to do is walk through what I'm calling here different levels of diversity. And when I discussed with a couple of you how to prepare this talk, it became clear that one of the things that would be relevant would be to show how genetic diversity and biological or phenotypic diversity, what's the interplay and what level of genetic resolution you see differences that we want to understand, but also differences that we don't understand, the drivers for those. So at the species level, of course, you're going to have different subspecies. This is something that I believe that has already been um, addressed, but this is a little bit dated. By a little bit, I mean a couple of years now, so it's not that bad. But on the left here, you have using multi-local sequence MLST data, multi-local sequence typing data. So this is essentially sequencing 7 genomes of each isolate. You have a summary of all the Xylella that is known out there. So you have these different subspecies that are widely accepted, a couple here that are in limbo a little bit. Pauca is actually much more diverse than this with the recent data that is emerging from Europe and Central America and South America for that matter, and fastidiosa, which is a stereotypical Xylella. Of course, there is another species in the genus now, recently proposed from Taiwan. We don't really know anything about the biology of that organism other than it causes disease in pairs. Um, so you have all this diversity here, and if you look at the genome tree on the other side here, which is with all the sequences currently available, the phylogeny really, it's pretty much the same. There's really not a whole lot of difference, and the groups that you see with MLST are, as with other bacteria, exactly the same groups you would expect to see with genome sequences. I want to bring up a couple of things here. First is that really most of the Xylella research has been done with about three isolates from this little group here. And this is Pierce's disease, primarily in California. There's a little bit of work done with this little group here from citrus in Brazil. And that's sort of pretty much it when it comes to biology. There's a lot of work getting started in Europe, but most of it is getting started for this group at large. But most of these other Xylella here, we really know very little about its biology. We know a little bit, but not very much. But the one little group here and one little group here is really where most of the information we have comes from in terms of biology. And this is just here showing for these genomes here, if you look at that same pattern in terms of number of genes, and here the number of genomes that you add to the analysis, just with 27 genomes, you already see that most genes that you find are actually unique to one isolate, while a core genome here is much smaller. So what we see with E. coli with 2,000 genomes, we're starting to see the same trend with Xylella, but at a much sort of smaller scale. Does this really matter? Are these different subspecies different in terms of their biology? We know genetically they form monophyletic groups, and the answer is yes. So this is some old work, but there's very little work that is kind of similar with large numbers for Xylella, whatever, half a dozen here is a large number, unfortunately. But you have here what we call a grape group, fastidiosa isolates from California. Here you have the multiplex almond isolates or group from California. And if you do cross inoculations, the almond one causes disease in almond, the grape one causes disease in grapes. However, the grape one also causes disease in almonds, almond leaf scorch here, while the almond one does not cause disease in grape. In California, you never find this genotype in grapevines or this group, period. While you do find some isolates that would be in this group here causing disease in almonds. So you do have, in this case in California, isolates going from grapes, from one subspecies causing disease in almonds and grapes, while the the opposite is not true. So you do have at the subspecies level, it's the easiest level to look at phenotypic and genetic diversity, and for some groups is well established, but the majority of those haven't really been studied yet. So as we go down this pyramid of different levels of resolution, the next group that I would cover, it would be a within subspecies. So there's a lot of interest in Xylella fastidiosa subspecies pauca in Europe, for example, or Xylella fastidiosa subspecies fastidiosa, which is the grape isolates from California fall within the group, and that was just found in Spain. So there's a lot of interest at the subspecies level. So the question is, do we have much diversity there, and does it mean anything? So we go from, so here, the best example for that really comes from Brazil. So if you've ever visited Brazil, in Sao Paulo area primarily, you have areas where you have a lot of citrus and coffee. And by a lot, I mean thousands of acres of continuous citrus or continuous coffee. Or sometimes you have a dirt road like this one right here, and you have, I don't know, hundreds of acres of citrus here, hundreds of acres of coffee here. You have Xylella diseases in both plants in Brazil, in this specific area, in fact. And you have, of course, the vectors are the same. They go from this tree to this tree, so on and so forth. So the question is, it's pretty obvious. Do genotypes from here cause disease here, and do genotypes from here cause disease here? So you do the typing, and okay, let's go back because this is incredibly busy here. But I have a point. So if you do the biology, you get these genotypes from different areas of Sao Paulo from citrus, you put them in coffee, you really get no disease. You get them from coffee, you put them in citrus, you really have no disease. And people have done this over and over, and the results are always the same. So let's walk through this very busy graphic here, and I do have a point. Let's start here from the two trees on the top. So let's start with this one here, B. So this is a phylogenetic tree with all the sequence types that were available until about a year ago when this analysis was performed. So here you have your different subspecies, fastidiosa, pauca. And let's focus on pauca, which pauca is what really is in South America. And the colored genotypes here are the ones that are actually present in South America. These other genotypes here are not present in South America. These are Central America and Europe. So what you have here is, doesn't matter if it's this tree or this tree, there are some differences on the trees, but you do have these groups here. What I want to illustrate is that no matter how you do your tree, you have these different groups. But if you just notice this one other tree here at the bottom, the limitations of what a subspecies is and what another one is not really clear. It's very arbitrary. So sandii down here at the bottom is this one little group, which clearly is very closely related to fastidiosa. Why this one here, for example, is fastidiosa and this one here is sandii is not really clear. In fact, if you get these two different genotypes here and you put them on this tree, you can't really see it, but it doesn't match. 61 is farther away from fastidiosa than 5 is. The other thing is because Xylella recombines, you do have problems of placement of isolates in phylogenetic trees. And essentially in this story here, you have, in South America, you have 3 isolates, 3 genotypes of multiplex, one over here, one over here, and one over here. They clearly represent 3 different introductions. You look at this other tree here, you have 2 that are close together and one that's out here, a gland, clearly different introductions from North America in this case. And you go over here in this other tree, you have one here, the other one is over here, and the other genotype is here. So multiplex is not native to South America. And in South America, it occurs in plum. It causes disease in plum. And it's an introduction, we think, from, it started in Argentina or Paraguay. We don't really know, but it's about 100 years old based on diversity and how much data we have. But so it's clear here, if you look at the trees, you interpret these and you have three different introductions. So far, so good. But then when you go here to the biology, essentially what we know so far is that the grape, the citrus group causes disease only. Citrus makes a phylogenetic group here. The plum isolates are over here, but they have actually exchanged genes with the citrus and green and blue are coffee groups. They have exchanged genes, and that's part of the reason that the phylogeny, the placement of these isolates come in different places in different phylogenies. It's because of recombination. So people think that you really have to sequence multiple alleles for properly place your genotype on a tree. Um, and despite the fact that you have gene flow from citrus to coffee and coffee to citrus, you still have these groups of citrus and coffee here, no matter which tree you look at, that are pretty robust. So you have here gene flow, you have the same vector, yet you have genotypes that are separate phylogenetically, and they're also separate biologically. I'm going to skip this. This is just to show that no matter how you analyze the data or generate data, we've done this for hundreds of isolates now, and the results are always the same. So you have diversity, you have these different subspecies, and it's very clear already that within subspecies, for systems that have been studied, you have very well group. So once you go to sequence types, which is at MLST level, you hear, I think Donato Boscia talk about ST53, which is the isolate in the sequence type in Italy, for example. We tend to think of sequence types as this small subgroup within subspecies. So let's look here specifically at California, which is only a couple of genotypes. So first, a couple of STs, I'm sorry. So here you have a bunch of different isolates, here and here. The details don't really matter. What matters is that we put these isolates into alfalfa as an indicator plant, and we looked at a very large number of phenotypic parameters or that we were able to measure in alfalfa. One of them was plant height. Another one was plant mass, which is with alfalfa, you can imagine it's sort of easier to measure than with grapevine, say. And you can see the amount of phenotypic diversity of these grape isolates from California, which again are really at the ST level in terms of their diversity. They're pretty much all the same ST. So you have genotypes here that cause 30% in reduction in plant height, and some that are much more pathogenic here, aggressive. You look at plant mass, which was how much you would harvest, and you can also see an extreme amount of phenotypic diversity in terms of virulence here, going from, the 100% would be controls, going from 30 to 90%. Over here, what you have is placement, or sort of phylogenetic placement, a genetic placement of these different isolates. The one on the right, please ignore. But it also shows that you do have at this subspecies, the sequence type level, you do have a fair bit of diversity. And here is just another way of representing phenotypic diversity here. So you have infection level, for example, and plant relative dry mass. And you can see that you have a lot of diversity in how much Xylella you have in a plant and the relative dry mass here. Of course, the more Xylella you have, in general, you have a more virulent or aggressive isolate. The point here is that you do have a lot of phenotypic diversity, and the question is, do you really have a lot of genotypic diversity? Since these are all pretty much the same sequence type? And the answer for that is yes. So you may have seen this from Sandy Purcell, I hope. Is that yes? Yes? Okay. I can't hear you. So I'm going to keep on talking.
Yes.
And,
yes, we've seen that.
Okay. So, so I'll skip through this. And essentially what we've done here is we collected isolates in about a 10-kilometer stretch of the Napa River. And we went to 5 different vineyards, and in each vineyard we collected about 15 different isolates, one each from a plant. Okay, so here you have a little vineyard, here you have the river, and here you have a plant, and we collected an isolate from this one and this one and this one, just randomly. So we brought them back to the lab and we did some genotyping. Long story short, what really matters is that we had about 93 or 95 different isolates, and we had 83 different genotypes. Okay, so essentially every single isolate that we got from a 10-kilometer stretch of Xylella from Napa was at the population level. These are all the same ST, but at the population level, they were different genetically. In addition to that, which was really cool, is that each one of these lines represent one isolate. So this represents one tree, one vine from one vineyard. Each one of these here is a vineyard. There are 5 vineyards. And what we try to do is you can, essentially, there are different approaches, and you can ask if you can assign an isolate. One of these lines here is an isolate. If you can assign it to a vineyard based on its genetic markers. Okay, so the question is, if you go to a vineyard here in Soda Creek and you get an isolate, and then you go and you look back to the entire population, can you assign that isolate to one of these populations? And theoretically, you would think that the Soda Creek isolates would be more closely related to the Soda Creek isolates, while the Silverado isolates would be more closely related to the Silverado isolates. Okay, these are different representations of that, the answer to that question. And what you would expect would be to see a solid color for each one of these different blocks. And what you see is exactly the opposite. So essentially, we could not assign a isolate to any vineyard because of how much diversity was already established in the area. So this, if you want to dissect this even further, you can ask, what happens within plants? So with a collaborator in Brazil, we ran this very similar experiment, and here we had 4 different citrus areas. In each area, we had a handful of plants that we got multiple isolates from. In reality, from each plant, we had 3 different branches, and from each different branch, we got 5 different pure clones at the end, okay? And the details of this are a little bit more complicated, but the point is that you run your genotyping at the population level with fast evolving markers, and you still find multiple, each one of these here, in some cases, each one of these was a different sequence, or not a different sequence, but it had a different fast evolving marker. Essentially, you have diversity at the branch level that, of course, serves as a source for natural selection later on. So if you have it here at this level, what happens if you get in one little colony here? Can you identify differences? And that would be within clonal lineages. So we've done a few different types of experiments here. And this is one that other people had done similar work, so it's not really surprising. But what we did is we just followed, we serially passaged Xylella from one plate to another plate to another plate, and you have P1, which is their first plate, and then Pn, which would be, you know, your other plate, right? This is a sequence of serial passages of one isolate. And here, as just you look at phenotype on plates, you can do this for 80 passages, and it changes pH of the plate. It's using different substrates. But more importantly is if you put it in plants, and here's the proportion of plants that were infected for P0, which would be your wild type, let's say, and an evolved lineage. And you can see that the trend here is downwards. Essentially, you can make Xylella lose pathogenicity with serial transfers, but you also have a lot of diversity here that's generated on plates themselves, and that leads to potential for selection, of course. So to end here and have some extra minutes for questions, I want to remind everybody that, of course, not all Xylella are made equal. And that is true all the way from species to isolate. There is an incredible amount of diversity, and all the biological knowledge we have really comes from a handful of isolates. There's been very limited work connecting genotype to phenotype. It's a major gap in Xylella research. Part of it is because it's a lot of work. It's not easy to do. It takes time. We know very little about how Xylella evolves and how it adapts. There is a lot of interest on that question right now, including ourselves, and I think in the. And one thing I really want to remind everybody is that there's literally a very limited amount of all the information is very few isolates and very few. So one of the things that's exciting is to see a lot more people working. I'm sure there will be new findings that are going to work being generated. But there are essentially 12 states in Brazil and 2 or 3 islands that are well studied until what's